Thank you very much for the opportunity to talk about this subject. I'm going to specifically focus on vertebrates in space and human studies and astronaut responses to the spaceflight environment absolutely inform and our motivation for these studies. I'm going to leave most of that discussion to Dr. Charles. Next slide, please. So I'm going to give you an overview. I'll be talking about fundamental questions in the context of vertebrates and some general comments about what occurs when the gravity vector changes in a multicellular complex organism, such as a vertebrate, what those early consequences are and subsequent. I'll be discussing the complexities because I thought one of an important component of this talk would be to help define what we've been able to learn and over the past few decades from animal studies and how to use that in our experimental design potentially for the station. I'll make a few comments about integrated physiology and the development of a ground-based model that helps inform. And as a final comment through the talk, you'll see some asterisks. Those refer to, I have a list of publications that very specifically addresses some of the points here, and they're quite selected. I think it's difficult in first entering this field to identify really the key references that are in a peer-reviewed available format. You'll be able to do that through emailing me. Next slide, please. So the fundamental questions, some of these are going to look remarkably familiar because they arise directly from some of the issues that the basic physics and biology that occurs at the cellular level. And the first question is, what are the organism's response to microgravity at the tissue level? And unfortunately, what scientists, biomedical scientists, and basic cell biologists have discovered is that sometimes responses that we observe in cultured cells, we may identify a specific molecule that plays a key role in a specific process. Sometimes when you go to the organism and disrupt that pathway, you find that there's a great deal of redundancy and there may be no effect, whereas in culture there may be large effect. So it becomes very important to look in the context of a more complex system at the tissue level. And the second issue is, what are the molecular and cellular as well as physiological mechanisms that account for the observed responses? Just because we measure a difference, it doesn't mean that difference is important. And what the mechanism, identifying the mechanism allows us to do is it lends a great deal of predictive power for other responses under other circumstances. Mechanism also informs countermeasures, ways to interfere with that. The third is, the third item is directly lifted from Neil's talk. What is the gravity threshold or the G dose dependence needed? We know virtually nothing about that at this time in the context of the multicellular organism, vertebrate. How does the microgravity modify the organism's response to other spaceflight factors such as radiation? And that is an area of research now, active research. And finally, what motivates the work with animals, and that is to use those studies to understand what the implications of microgravity responses are for human health and the development of countermeasures. This is relevant both to space and on Earth. Next slide, please. So upon entry into space, two things happen immediately, very acutely. That is, there's a headward shift in the body fluids, and there are rapid compensatory changes that take place in the circulatory system. Also, the one attribute we think about most often is that we're exposed to weightlessness. So from the 1G environment that we've all evolved in, we enter an environment in which we no longer experience mechanical loading in in the native form. We think of that most often in terms of the musculoskeletal system, the atrophy that occurs with muscle, and the bone loss that occurs. But in fact, all cells may be affected to some extent, and more work is needed to define that. And finally, an immediate disorientation that results from these changes. Now, these are acute, and acute changes don't necessarily lead to long-term changes, but in fact, of course, we know they do. Next slide, please. This slide is basically to communicate a couple points. One is that after this acute phase, our bodies have this amazing ability to adapt to changes, and as we all know now, we survive quite nicely in space for quite a long period of time, and we adapt quite well. But in order to understand the underlying mechanisms and how to go in and disrupt that process, it's important to understand what are the underlying structural and characteristics. By structural, I mean both the composition and the anatomy of a given change, because that's very important, because once in a given tissue that responds to that acute change, once there's a structural change, then return to Earth it may be profoundly affected, and it may trigger subsequent changes. The next point I'd like to make about this is that these physiological responses, as scientists, we all tend to study them in a specific system, but in fact, they're all quite interrelated. And finally, and this has repercussions for analyzing flight data once it's obtained, is that these responses are time-dependent and different for each system. Typically, in flight experiments, we get a snapshot of one or several time points after flight, and that, so that we may not appreciate some of the changes that are occurring acutely elsewhere, but in terms of human health, it's important to start to see that as an integrated whole. Next slide, please. So in terms of practical aspects, there are complexities to the space environment. I'm only going to mention a few because of the impact on interpretation of previous results and thinking about experimental design for future experiments. There are some variables in space, some independent variables, many of which tend to be rather poorly controlled, or at least we wish they were better controlled. Each has the potential, each of these variables has a potential to profoundly affect the function of a multicellular organism, and yet we don't know how important that is until it's tested or looked at in the context of other studies. The other aspect of these poorly controlled variables is that it can complicate between and within experiment comparisons. So we have to interpret previous results with great care. The first studies that were done with animals, extensive studies that were done with animals with US investigators were done on the Cosmos missions, and the launch and landing profiles were quite dramatically different, and housing conditions were quite dramatically different. And so all of the results that obtained in one study may not be obtained in the other study, but in fact, there's value to this, in that together, when we see this, we can we are encouraged that this is a physiologically and biologically significant change. I just want to mention quickly a couple aspects. The unique aspects of housing. Some of these things we don't think about on Earth. Rodents, in particular, as an example, are exquisitely sensitive to housing density. We probably all remember from high school learning about putting too many rats in a cage. In fact, what happens in space is now the two dimensions of the floor space turn into 3 dimensions, and we don't have a lot of we don't have a database in which to know how to interpret that change, but it needs to inform how these cages are designed. Next slide, please. So what are the differences that arise specifically in the course of the experiment? Launch and landing, there are transient acceleration and vibration forces that have the potential to influence. And I want to mention one important study in terms of landing. Most of the vertebrate studies have been done on samples that have been recovered after animals have been exposed to space flight and then returned to Earth. And there is a whole suite of changes that occurs in skeletal muscle in response to flight, atrophy, which you're probably familiar with, but also there was evidence of edema, accumulation of fluid within the tissue, and some lesions in the sarcomeres. And what they found through studies that allowed, that facilitated in-flight dissections and sample collection was that the changes, the damage to the muscle arose due to landing. Atrophy itself was a unique consequence of the weightless environment of space. And then other issues, of course, of timing. Now, fortunately, many of these factors and variables that I've been talking about can be tested and are tested in control experiments, and that really necessitates for PIs to have a pretty robust ground-based research program to support those control studies. Next. So NASA has flown a wide variety of vertebrates at different stages from embryonic to juvenile or adult. The vast majority have been rodents, mostly rats initially, and now more and more mice, which have the advantage of being able to manipulate them genetically. Some of the factors that have come up, two of the factors that are important to consider is stress and age of the animal. Our astronaut population is adult, but many of the earlier studies were done with growing animals. Nonetheless, if one of our goals in terms of the basic science and understanding what's going to happen in the future with exploration, if one of our goals is to understand cradle to cradle development in a microgravity environment, we need to understand these other developmental consequences of being in space. Next slide, please. So, as in any scientific investigation, what's key is selection of controls. And I want to just briefly mention the different kinds of controls that you can think about in terms of trying to define the influence of microgravity on vertebrates. So typically in flight experiments, I'll talk mostly about rodents from now on, in flight experiments, there are typically two kinds of cage controls, one that are typical standard cages that we use in our ground-based studies, and the other is the flight hardware cages, because animals, and in particular mice, are exquisitely sensitive to changes in things like position of food bars and wire versus flat cages. So it's important to have it in the course of the experiment, those appropriate controls. Now, the second item listed under that top bullet, onboard centrifuge, there's been very limited opportunities for that. There was in the Cosmos mission an onboard centrifuge that was used in an effort to understand what might be the effects of microgravity on rats, those animals having been exposed to all the other stressors and changes in spaceflight. And those studies provided some initial evidence that the bone changes and some of these changes did not occur if an animal was exposed to 1G, but on a centrifuge. But much more work needs to be done in that area. The second way this is done in a practical level is time delay of controls. So the ground controls can be euthanized or samples collected 2 days later or 3 days later so that you can match precisely the conditions of flight. And the other that I want to comment on, because this is a question that comes up often, is, well, if you take an animal in a rodent in space, how much of that response is due to stress and how much is actually due to microgravity or the fluid shifts? And so spaceflight experiments subsequent to the initial studies where you define the effect are useful because you can address that. So one of the ways that was addressed in the past with rats is that the stress hormone corticosterone, which we have the analog is called cortisol, which we produce when we're stressed. These animals were manipulated to maintain constant stress hormone levels in space and to prevent any rise that would occur with stress. And in those animals, they still had bone loss. So we knew from that the bone loss is independent of the stress response. We know less about those kinds of studies need to be done also in other species. Coupled with physiology, and when that's been done in the past, despite the limited number of flights we have, it's held up pretty well. The molecular gene expression changes overall have pretty well reflected some of the catabolic changes in the musculoskeletal system. Now, the other way you can accomplish an integrated approach, much greater than any single investigator alone, is how the animal spaceflight experiments are conducted themselves. Next slide, please. That's done with basically with the tissue sharing approach, and I'm going to show you a couple examples of that. So SLS-1 and SLS-2 were flight experiments, and there's extensive tissue sharing. I'm just showing you a few of the areas. So by having other investigators analyze samples, we're able to get a bigger picture. Next slide. The Neurolab studies are an example where a flight was, the studies were dedicated to a single system, the neurological system. And also in this study, there was in-flight dissections. So as we all know, our opportunities are extremely few and far between, and are limited by payload size and many other variables. And so to make progress in understanding the underlying processes, physiological processes that occur, we need to be able to do basically iterative experiments, learn from one study, and then turn around, repeat that result, and modify the experiment so we gain new insight. And that's how we make rapid progress. That's how drugs are discovered on Earth. And, but that's quite limited in space. So next slide, please. About 30 plus years ago, a ground-based model was developed to facilitate that. That's the Highland unloading model. This is where rats or mice can be unloaded in special cages that allow them free movement. And what we find in these animals is that there is a headward fluid shift, as we see in the astronauts, and also there's also selective unloading of the hindquarters. This model has enabled over the years an extensive analysis, not only of how this disuse affects specific systems, but how recovery occurs and how might we accelerate recovery post-flight. And also, it's an excellent model in terms of animal model for clinically relevant conditions such as prolonged bed rest and inactivity. Now, in rats, we found many of the changes that we see that occur in astronauts and in animals in space also occur in rats with this model. Not all, so it's important to be critically evaluate them, but they are entirely consistent. In mice, there's also consistency in the responses. They've been studied less in space, as I mentioned, but some of the data suggests that stress may play a more important role in the mouse than the rat. Next slide, please. So for concluding comments, our animal experiments that have been done over the last 30 years in space have first defined some of the main effects of microgravity on the vertebrate, and also just begun to define mechanisms and countermeasures. But all of these studies have been done really in short-term flights. We find ourselves in a really unique situation. That is where humans have been in space for as long as 2 years, for very prolonged periods of time. Our experimental animals that on Earth we use to ensure safety and of drugs and long-term consequences of a given treatment, those animals have only been, vertebrates have only been in space up to 19 days on the shuttle in terms of results obtained so far. And so I see the ISS as a really unique opportunity potentially to do both the basic cell biology and translational research that will help move this field forward. Thank you. Yes. There have been extensive, I'm sorry. The question is, have there been studies in the immune system and the immune response, either animals or astronauts? And the answer to that question is there have been extensive studies, and it's a very active area of research. The studies in astronauts previously have shown that if you take the blood cells, the immune cells, out of the astronaut and challenge them, there's a defective response. And in fact, when investigators have looked at the immune system in rats and in mice after exposure to space, there are a complex series of changes that occur. And the question is whether those are stress or unloading, and I think the answer to that question would depend on who you ask, which investigator. Some of the changes are seem to be very clearly related to stress, but there's some good, solid, and brand new evidence that should come out soon that the results obtained in cell culture in terms of astronauts' blood actually have an influence at the whole vertebrate level, the whole system level. Anecdotally, there's always been reports of more impaired wound healing, skin lesions, and also activation of latent viruses and infections, upper respiratory infections. So this is a really important area of research, especially in light of the newer evidence that there's greater virulence. Yes, sir. Right, and the question is, are satellite cells able to repair the damage in skeletal muscle that occurs? Is there damage repair? Is that, or does a muscle fiber repair itself? I believe that both play a role. I think that some of the work in terms of replacement by satellite cells is relatively new work that's being done, but there are definitely repair processes that occur. It does much like in bone. After flight in animals, the repair processes are not complete. especially in those animals that have experienced damage with landing. Yes?
The ramping of fluid for entry for the shuttle and station crew members, does it vary? What's the difference on it?
Could you define what you mean by ramping of fluid?
They ramp up on fluids and electrolytes before they re-enter.
Mm-hmm.
What's the difference
On the station versus, I'm going to leave that to Dr. Charles to address. It helps address the changes in the compensatory changes in the circulatory system before the gravity transition. But I don't know in terms of differences between the station and the shuttle. Someone want to address that?